Good day, everyone. I welcome everyone to upcoming webinar on PD-1 blockade cancer immunotherapy, current issues and perspective, which is going to be presented by Dr. Alok Kumar from Kyoto University, Kyoto, Japan. Dr. Alok Kumar has completed his PhD studies in cancer immunotherapy under Professor Tasuku Honzo at Graduate School of Medicine, Kyoto University, Kyoto, Japan. And Dr. Alok joined Kyoto University in the year 2015. Professor Tasuku Honjo discovered PD-1, an immunoinhibitory receptor on immune T cells, for which he got Nobel Prize in Medicine in 2018, along with Dr. James P. Allison. Here in the picture, you can see Dr. Alok with Professor Honzo. He celebrated the moment when Professor Honzo got Nobel Prize in 2018. You can see the celebration picture over here. And Dr. Alok belongs to Bihar state in India after completing his schooling and bachelor study in biotechnology. He qualified many entrance tests for his master's study, and finally in 2011 he joined the very prestigious Indian Institute of Technology Roorkee for MSc Biotechnology study. He is an editorial board member for Gnome Publications, and we are very proud to get associated with Dr. Alok Kumar. And here he will be presenting his upcoming webinar on PD-1 blockade cancer immunotherapy, current issues and perspective. I'm now going to share my views, and I'm going to hand over the webinar to Dr. Alok Kumar. Dr. Alok, you can go ahead.
First of all, thank you so much for the nice introduction, and thank you to the whole committee member of Genome Publications for giving this, for organizing this webinar and giving me opportunity to present my work and give some insight about PD-1 blockade therapy and the recent advancements ongoing in PD-1 blockade therapy. And thank you to the listeners as well for joining us.
Yeah, you're welcome.
Uh, so can you see my slides presentation?
Yes, I can see your slides.
Uh, so the title is PD-1 blockade cancer immunotherapy, current issues and perspective. Just wait.
Yeah.
Yeah. So I will cover two topics in this talk. The first is rationale of PD-1 blockade therapy. So here I will discuss how PD-1 blockade therapy works. And second, PD-1 blockade therapy, actually some patients still show unresponsiveness. So I will discuss why some patients are unresponsive. And the topic title will be mechanism of unresponsiveness to PD-1 blockade therapy. So first I will start with cancer statistics. According to IHME 2016 report, cancer is the second most leading causes of death worldwide, and around 18.1 million new cancer cases were detected in 2018, according to IARC report. And the mortality cases were around 9.6 million cancer cases. So these statistics show the severity of cancer worldwide and demands for a treatment, treat, better treatment and very improved methods of cancer treatments. And as per therapeutic strategy we consider, so there are four modalities of cancer treatment. The first is surgery, which is well known from very long time. The second comes radiation therapy, and it came into practice around 1900 after discovery of X-ray and following. Radiation therapy became popular around that time, around 1900. The third therapy is chemotherapy, and it came into practice around 1920. And that time, a plethora of chemical compounds, chemicals were screened and formulated for targeting the cancer cells. The last one is immunotherapy, and it came into practice around 1960. And in immunotherapy also, there are different modes of treatment. The first is non-specific immune stimulation. Here, by using some cytokines or some other stimulatory factors, we stimulate the immune cells non-specifically. For example, IL-2, interferon gamma, like that. Second is adoptive cell transfer. Here, the uh, first we isolate some cancer targeting immune cells from the patient body, and then we engineer in the in vitro system, and further after proliferation and expansion, we introduce back to the patients, cancer patients. The third mode is vaccination study, and fourth one is immune checkpoint blockade. So here I will discuss immune checkpoint blockade in detail and how it works. So before going in detail about the cancer immunotherapy, first we should understand how the cancer develops and a concept known as cancer immunerating, how it works, how the cancer develops even in the presence of immune system. So the immune cells have capability to detect the cancer cells and kill them. And this concept is known as cancer immunosurveillance. And this concept was first given by Bernard and Thomas in 1957. However, there was some issue, and still cancer, but the major problem was that why cancer cells are growing, even if even the immune cells can detect them and can kill them. So to answer that question, in 2002, Professor Robert Silver gave an updated concept, and it's known as cancer immunoediting. That is immunosurveillance to tumor escape, and it consists of three stages. The first is elimination, second equilibrium, and third is escape. In elimination, the immune cells like NK cells and CD8 T cells will detect and will kill the cancer cells, the cancer initiating cells. Still, some cancer cells will still survive, and they will grow further and they will proliferate. They will develop some oncogenic more mutations, and a situation will come where the immune cells will detect and the cancer cell also will proliferate vigorously. So this will be a kind of equilibrium, and so here the immune cells, the cancer cell will be more immune selected, and in last the cancer cells will develop will use different immune suppressive mechanisms, and finally it will escape the immune surveillance. So in this way the cancer cells develop even in the presence of immune system. This is the CD8 T cell fate during immune response. So first, during antigen exposure, the antigen presenting cells present the antigens to the T cells in the draining lymph node, and the particular draining particular CD8 T cells will proliferate, will be activated, and it will proliferate, and it's known as effector T cells. Here, most of the effector T cells will now go to the inflammation site or tumor site, where they will detect and kill the cancer cell. And during this process, most of the effector T cells will be, uh, just wait one moment. Most of the effector T cells will be died because they are the terminal differentiated T cells. And during this process, some of the effector T cells will change into memory T cells, and that have the long life, and that will counter the second infection. So here during the T cell activation, mitochondria play important role, and mitochondrial in the electron transport system, it generates reactive oxygen species that will help in the T cells activation. And during the effector phase of T cells, it develops it depends mainly on the glycolysis for their energy production, while the memory T cells depends largely on the oxidative phosphorylation and fatty acid oxidation for their survival and for their energy demand. In the host body, immune system is immune responses are in very controlled is a very controlled process, and this is known as immune homeostasis. This is the balance between immune activation and the immune suppression. If immune stimulations are more, that in that case the T cells will be highly active, and that may cause the autoimmunity. While just opposite, if immune suppressions are very higher, that then in that case the T cells will be deactivated, and that happens that is called as tolerance, and it happens during chronic infection and cancers as well. So first, during the immune response, the antigen presenting cells present the MSC loaded antigen to the T cells, and that T cell receptor TCR, we call in short, detect this MSC loaded antigen, and this gives the first signal to the T cells. Now the T cells will be activated or it will be suppressed. It depends upon the second signal here, the second signal. So this second signal may be a stimulatory, that is, it will give the positive signal, or it can give even the negative signal. So here the positive, that is the stimulatory signals are like ICOS, 41BB, ICOS, and CD8. These are the positive stimulatory signals, while PD-1, CTLA4, these are the negative, that is the inhibitory signals. So the T cells will be activated or depend or suppressed. It will depend on these secondary signals, second signals. Now I will talk about how the PD-1 works. PD-1 is a program cell death protein 1, and it is an immunoinhibitory receptor expressed on activated T cells. And its ligand PD-L1 expressed on antigen presenting cells or the tumor cells. And the ligation of PD-1 to PD-L1 gives negative signal to T cells, and the T cells will be deactivated. If we block this signal by using either PD-L1 antibody or PD-1 antibody, this signaling will be blocked and the T cells will be again activated, and the T cells will kill the cancer cells. By using this strategy, the T cells will be reactivated and we can move back this process towards the immunosurveillance. That is, we can cure the cancers. This is one of phase 3 clinical trial data. Here, the melanoma cancer patients were treated with either a standard chemotherapy, dacarbazine, or PD-1 blocking antibody, nivolumab. And after 1.5 years, the survival was around 72% for PD-1 blockade therapy, while it was just 20% for standard chemotherapy. And this is another clinical trial data. Here, the cancer patients were divided into different groups based on mismatch repair proficient and deficient. If the tumors are mismatch repair deficient, then in that case, the tumor cells will accumulate more and more mutations. And here you can see that the black and the blue dots related with mismatch repair deficient, and you can see here the tumor were rejected by PD-1 blockade therapy, while the patients with mismatch repair proficient colorectal cancers, they were not benefited with PD-1 blockade therapy. So this is very important properties of PD-1 blockade therapy, that the PD-1 blockade therapy will work more when the cancer stages will be higher, like stage 3, stage 4. That time PD-1 blockade therapy will work more, because that time they have accumulated more and more mutations. And so other advantages of PD-1 blockade therapy are that it has less adverse effects, sustained effects to responders, and it is effective for a wide range of tumors like melanoma, renal cell carcinoma, and many more. And FDA has recently approved it for mismatch repair deficient tumors and microsatellite instability high tumors as well. This is the list of FDA approved immune checkpoint blocking antibody. So for PD-1, there are 2 antibodies, nivolumab and pembrolizumab, and other 3 antibodies are blocking PD-L1 on the APC or cancer cells. This is PD-1 chronology, and this is a very long history, and it started back in 1992 when my supervisor, Professor Tasuku Honjo, discovered this molecule PD-1 at Kyoto University. And after that, the first PD-1 knockout mice were generated in 1999, and in 2002, the PD-1 knockout mice has shown that they have the cancer effect, anti-cancer effect. And the ligand PD-L1 and L2 were discovered in 2000 and 2001, respectively. And the first clinical trial were performed using PD-1 antibody in 2010. And after following years, each and every years almost, The FDA has approved different blocking antibodies for the cancer treatment. And finally, in 2018, Professor Honjo, along with Dr. James P. Ellison, got Nobel Prize in medicine for this novel cancer immunotherapy work. Despite great success of PD-1 blockade therapy, still there are some issues that need to be answered. First, a significant fraction of patients still show unresponsiveness to PD-1 blockade therapy. So, what is the mechanism of their unresponsiveness? And second, what is the best combination therapy to rescue less responsive cancer patients? To understand the mechanism of the unresponsiveness, we used mouse model here. And first, we screened which uh, which mouse cancer cell line is responsive and which is unresponsive. So first here we injected tumor cells on day 0, and after every fifth day we injected PD-1 blocking antibodies, and we measured the tumor size. Here this is the tumor growth of colon cancer MC38, and here this is for glioblastoma GL261 cell line. The open circle denotes for the control IgG, and the black circle denotes for PD-L1 treated group. And you can see here that the tumor was rejected after PD-1 blockade therapy. So this shows the responsiveness. It means the MC38 is responsive tumor cell lines, and the same for glioblastoma GL261. While just opposite, when we injected melanoma, that is the B16, or lung cancer LLC, we found that the tumor was not rejected. It means that they are unresponsive. So uh, using this schedule, I screened many different cancer cell lines from both backgrounds, C57BL6 and bulb-C background. And here the responsive tumor cell lines are marked here in red color, and the unresponsive tumors in blue color. Further, we were interested how the immune responses were different in the host bearing responsive tumor or unresponsive tumor. So we followed this schedule, and here we injected responsive tumor MC38 and GL261, or unresponsive tumor B16 and LLC on day 0, and we performed 2 doses of PD-1 blockade therapy, day 5 and day 10, and we sacrificed mice on day 12, and we harvested draining lymph node and the tumor mass as well. Now this is the DL analysis. The CD cells were gated, and they were plotted based on the intensity of CD62L and CD44, which are adhesion marker and activation marker respectively. And we plotted, this P3, that is the effector memory, are now they are ready to go to the periphery, and they are in the tumor mass. They will detect and kill the cancer cells. So we plotted this effector memory populations, and we found that the absolute number were significantly higher after PD-1 blockade therapy in the host bearing responsive tumor, but there was no change in the host with unresponsive tumor after PD-1 blockade therapy. So it means that the PD-1 blockade significantly enhances the number of effector CD T cells in mice with responsive tumor. Uh, Further, we analyze the tumor mass as well. So here, this is the tumor infiltrating lymphocytes that kill the cancer cells. These are the CD T cells, and we found their frequency were higher after PD-1 blockade therapy in the host with responsive tumor, but there was no significant change in the frequency after PD-1 blockade therapy in the host with unresponsive tumor. And for the effector functions, we stained with interferon gamma intercellularly, and we found that the interferon gamma expression was higher after PD-1 blockade therapy in the host bearing responsive tumor, but there was no change in the host with unresponsive tumors after PD-1 blockade therapy. So this suggests that the PD-1 blockade significantly enhances the number and effector function of tumor-reactive CD T cells in mice with responsive tumor. Further, uh, we we reported that mitochondria are activated in PD-1 blockade condition. This is the RNA-seq data of CD T cells from lymph nodes, and you can see here that the mitochondrial activation associated genes were highly upregulated in PD-1 knockout mice compared to wild-type mice. So, based on this finding, we measured the mitochondrial respiration here using seahorse machine On the left panel, this is electron transport system, and the electron transfers through different complexes, and finally, the electron accepts oxygen accepts this electron, and this uh, this makes an hydrogen and concentration gradient and ATP produced. Now, in the seahorse machine, we use different chemicals to measure this oxygen consumption because there will be more oxygen consumption, so it means there is more oxidative phosphorylation. So, oxygen consumption is directly proportional to electron transfer chain reactions, or you can say the mitochondrial respiration. So, here in this machine, we measure this oxygen consumption. So, first oligomycin chemical will be added, and that so the oxygen consumption will be reduced. In the second stage, FCCP chemical will be added. FCCP is uncoupler that will block, that will disturb this hydrogen and concentration gradient, and so there will be maximal oxygen consumption rate. And finally, the two chemicals, rotenone and antimycin, will be added that will completely shut down this electron transport chain system, and we will get non-mitochondrial respiration. And when we will subtract the basal respiration from maximal respiration, we get spare respiratory capacity. And this capacity denotes how long the cell can survive in stress conditions. If this spare respiratory capacity is higher, that cells can survive longer in stress conditions. So using this machine, we measured the oxygen consumption rate from the CD8 T cells from different groups. Here, this is for MC38, and the open circle denotes control IgG group, and the black circle denotes anti-PD-L1 group. Here you can see that the oxygen consumption rate were higher in PD-L1 treated group, and this happens mainly in the host bearing responsive tumor, while there was no change in the host with unresponsive tumor. And we calculated we calculated the other parameters as well, like basal respiration, spare respiratory capacity, and ATP turnover also, and they were higher after PD-1 blockade therapy only in the host bearing responsive tumor, but there was no change in the host bearing unresponsive tumor. So far I can summarize the findings up to here in one sentence, that PD-1 blockade causes increase in number, effector functions, and mitochondrial activation of CD8 T cells in the host with responsive tumor, but not with unresponsive tumor. Now we'll move to the uh, how to understand the unresponsive mechanism. So to understand the unresponsive mechanism, first we need to understand the cancer immunity cycle. Here the cancer antigens will be presented to the T cells in the draining lymph nodes, and the particular CD8 T cells will be activated, and they will expand more, and they will move to the periphery, and from there it will go to the tumor site, and will there it will detect and kill the cancer cells. So this is the cancer immunity cycle. And for active immune responses, this cancer immunity cycle should be circulated very smoothly. If there is blockage at any of these pathways, that will lead to the unresponsiveness. So it's possible that there is a blockage here in the uh, like CD8 T cells and tumor cell interaction, or it's possible that any this immunity cycle has been blocked systematically at any stage, and that block the CD8 T cells activation and functioning. So broadly, we divided the unresponsiveness mechanism into two groups. The first is that local immune ignorance, that is CD8 T cells and tumor cell interaction, that is their detection by the host immunity, and second is that systemic immunosuppression. To understand the immune recognition, here we compare the tumor growth between wild type and reactive knockout, that is immune compromised mice. We injected responsive tumor MC38 and GL261, and here we found the open circle denotes wild type, the growth in wild type, and the black circle denotes growth in reactive knockout, that is immune compromised. And you can see here clearly that the tumor growth was much faster in reactive knockout, that is immune compromised, while the tumor growth was much slower in wild type mice. It means that the immune system are recognizing these cancer cells, and that's why they are trying to kill that cancer cells. So that's why here the tumor growth is were lower. So it means that we can say that the responsive tumors are well immune recognized. While on the other hand, when we injected B16, that is melanoma cancer cell, we found that the tumor growth were almost same in rat to knockout mice and in wild type mice. It means that they are not recognized by the host immunity. While for LSU, we found that they are little or less immune recognized by the host immunity. Now to understand the second aspect, that is the unresponsiveness based on systemic immunosuppression property, that is the SIP. For this, we divide a novel bilateral tumor model, novel strategy. Here in this one host, we injected both tumors. So first unresponsive tumors were injected on the left side, and we injected responsive tumor on the right side, and we performed PD-1 blockade therapy. And after PD-1 blockade therapy, there will be two cases. If that responsive tumor will be rejected, then in that case, the unresponsive tumor on the left side, we can say that they don't have immunosuppression. It means they are unresponsive because of their local immune ignorance. They are not inhibit the immune responses systemically. In other words, we can say that. Now, the second possibility is that this responsive tumor will not be rejected by PD-1 blockade therapy. So we can conclude that the unresponsive tumor on the left side has some systemic immunosuppression, and that's why that systemic immunosuppression did not allow the functioning of PD-1 blockade therapy, and that's why the responsive tumor did not reject. Or in other words, we can say that some soluble suppressive factor exists because of unresponsive tumors. So based on this hypothesis, we try to classify or we try to characterize the different unresponsive tumors. So using this schedule, first we injected here unresponsive tumor B16 or LLC, and when the tumor size was around 60 to 70 mm around day 6, we injected responsive tumor MC38 on the right side. And we started therapy on day 8. And after every fifth day, we injected PD-L1 antibody, and we measured the tumor size of MC38 on the right side. And what we found was very interesting. When B16 was present on the left side, the tumor size of MC38 on the right side were reduced by PD-1 blockade therapy. So it means that the PD-1 blockade therapy worked here, and that's why the tumor was rejected. So we can say that the B16 does not have immunosuppression here. While the situation when LLC was present on the left side, in that case, the tumor size of MC38 was not rejected by PD-1 blockade therapy. It means that the PD-1 LLC has created some systemic immunosuppression in the body, and that's why the PD-1 blockade therapy did not work, and the MC38, the responsive tumor, was not rejected. So we can say that the LLC is systemic immunosuppression positive, means SIP positive, and B16 is SIP negative. There are some reports that the cancer cells lose MSC class 1 molecule to evade from the immune recognition. So we tested that possibility here for B16 because so far here we know that B16 does not cause any systemic immunosuppression, and they are not recognized by the host immunity. So we tested this MSC class 1 molecule on their surface, and here you can see that the MSC class 1 expression was enhanced in PD-1, MSC class 1 molecule expression was enhanced after interferon gamma stimulation in this MC38 means responsive and the unresponsive LLC. But there was no change, or you can say means there was no MSC1 expression in B16. So we can say that the loss of MSC class 1 could be the reason of unresponsiveness for B16. So this is the summary of unresponsive tumors. So this is the summary for both backgrounds, C57BL6 and BULB-C. So for B16, we can say that it does not release any suppressive factor, while for activation of acquired immunity, it does not incite the acquired immunity. And LLC, yes, it releases suppressive factor according to our bilateral tumor model, and less activate the acquired immunity. And PAN02, yes, it releases suppressive factor, and yes, it does not incite the acquired immunity, and the same for CT26 and VI3 from bulb C background. Further, we were interested how this immunosuppression by ALC inhibit the immune responses against the responsive tumor. So, using the same schedule here, we injected unresponsive tumor, that is the SIP negative and SIP positive, that is B16 and LLC respectively. We injected on day 0, and on day 6, we injected MC38, and we performed two doses of PD-1 blockade therapy. And on day 14, we sacrificed mice and we harvested brain lymph nodes. And further, we analyzed. So you can see here that when B16 was present on the left side, uh, so after PD-1 blockade therapy, the T cell number were increased on the right side, that is towards the MC38 side. So it means that the B16 does not create any systemic immunosuppression, and that's why the PD-1 blockade therapy worked, and we got significantly higher cell number, total DLN cell number. While when LLC was present on the left side, we can see here that there was no significant enhancement or change in total DLN cell number after PD-1 blockade therapy. So it means that the LLC is creating some systemic immunosuppression in the body, and that's why PD-1 blockade therapy did not work. Now, we were interested how this suppressive factor from LLC inhibit the mitochondrial respiration, because previously we have seen that the mitochondria was enhanced after PD-1 blockade therapy in the host with responsive tumor. So here we measured the CD8 oxygen consumption rate of CD8 T cells from the DLN cells that were collected from the outside, and the black circle denotes the PD-L1 treated group, and the open circle denotes control IgG group. And we can see here that the oxygen consumption rate was higher when B16 was present. B16 was present on the left side after PD-1 blockade therapy, but there was no change after PD-1 blockade therapy when mice had LLC on the left side. And we calculated different parameters like basal respiration and ATP turnover, and we found that these parameters were also significantly higher when B16 has on the when the mice had B16 on the left side. So it means the B16 does not cause any systemic immunosuppression, and that's why PD-1 blockade therapy worked, and the T cell number were also increased, and the mitochondrial respiration was also enhanced. But there was no change when mice had LLC on the left side. So far, I can summarize the conclusion. So conclusion one is that responsive tumors are recognized by the host immunity, while unresponsive tumors are not or less recognized by the host immunity. Second, unresponsive tumors classified into two groups based on the property of systemic immunosuppressive property, that is SIP. So SIP positive LLC and SIP negative B16. SIP positive causes humoral immune suppression that could be the reason of their unresponsiveness, for example, LLC. And third conclusion is that SIP negative tumors rely on local immune ignorance to evade immune recognition or surveillance, for example, B16. Now, to understand the suppressive factor from LLC, what are that? What are those factors? So first, we harvested the culture supernatant from different cancer cell lines like B16, LLC, and others, and we tested their effect on the T cells proliferation in our in vitro assay. Here, the T cells were stimulated with CD3, CD28 antibody for the TCR stimulation, and we added a culture supernatant, and we stimulated the cells for 48 hours. And after 48 hours, we measured the cell proliferation using thymidine incorporation. We can see here that when there was no CD3, CD28 stimulation, there was no peak. The T cells were not proliferated. When there was no supernatant added, we got higher peak. We got some peak means positive, around 8,000. And when B16 supernatant was added, the value were almost same. It means that the B16 does not release suppressive factor. While surprisingly, we found that when LLC supernatant was added in that well, we found that the proliferation was very significantly inhibited. So it means that the LLC is releasing some suppressive factors that inhibit the proliferation of T cells. And if this LLC contains some suppressive factors, so their effect of suppression should be diluted when the culture supernatant will be diluted serially. So first we diluted the culture supernatant from B16 for the control group and LLC here, and the pre-diluted culture supernatant were added in the well in the same protocol. And further, after 48 hours, we measured the T cell proliferation, and we found that when we diluted the culture supernatant from LLC, the suppression effect gradually lost, and the T cells proliferated more and more. It means that the LLC, so this further confirms that the LLC is releasing some suppressive factors, and while the B16 does not release any suppressive factors. Further, we were interested, so what is the nature of that suppressive factor that were releasing from LLC? Whether it is proteinaceous molecule or it is some small molecules, non-proteinaceous in nature. So if this is protein, so that protein will be denatured after boiling treatment. So after boiling, the protein nature will be degraded and the suppressive effect will be gone. And second, if this is a small molecule, so here we treated with DCC. DCC means dextran coated charcoal. So dextran coated charcoal treatment will absorb all the small molecules on their surface, and so the supernatant will be free from small molecules. So we tested this hypothesis, and we found that the boiled supernatant has almost the same suppressive effect as the parent supernatant. It means that they are not protein. They are not proteinaceous in nature. While we found that the disease treatment supernatant has no suppression, means most of the suppression have effect has gone. It means that these suppressive molecules in the LLC supernatant are non-proteinaceous in nature. They are a small molecule. Further, we were interested what is the size of that small molecule. What is the size, like 1 kD, 2 kD, 3 kD, like that. So using these different filters, we fractionated the culture supernatant of LLC into different fractions, like less than 3 kDa fraction and more than 3 kDa fraction. And we found that the 3 kDa fraction has almost the same suppressive effect as total supernatant. So it means that majority of the suppressive factors comes in this range, means less than 3 kDa fractions. And when the 3 kDa fractions were dissociated, means when the treatment will absorb all the molecules, small molecules, the suppressive effect gone and proliferation was increased. So it means that, so we can conclude that the suppressive factor is non-proteinaceous in nature. They are a small molecule of size less than 3 kDa. Further, we were interested from the mitochondrial respiration, from the mitochondrial activation point of view, and here also we measured the mitochondrial respiration. So the open circle denotes without CD3 CD20 stimulation, and the black circle denotes CD3 CD20 stimulation when there was no supernatant added. That is the control group. So we got some higher peak here. And when B16 culture supernatant was added, that is the open square, we found almost same as no supernatant. It means that the B16 does not have any suppressive factor. Well, surprisingly, we found that when LLC supernatant was added, we found that the oxygen consumption rate was very severely reduced when LLC supernatant was added. And in the same experiment, we measured oxygen ECER, that is extracellular acidification rate. This value tells about the glycolytic respiration, and we found that even the glycolytic respiration was also very significantly inhibited by LLC suppressive factors. And this is the two variables graph. So on the y-axis, this is OCR, that is oxidative phosphorylation, and on the x-axis, this is ECR, and it tells about glycolytic respiration. And we can see here that when we added LLC supernatant in the well, both oxidative phosphorylation and glycolytic respiration was very severely inhibited compared to no for B16. So in summary, we can say that the reduction of mitochondrial glycolytic respiration by suppressive factor from LLC, but not by B16. Then we further performed experiment to understand that whether this suppressive factor act directly on the mitochondria or whether it is functioning like transcription translation by this process. So to test that hypothesis, we stimulated T cells in the presence. We just added culture supernatant and just for the short time, that is just 2 hours. And there was no CD3, CD28 stimulation. And after 2 hours, we measured the mitochondrial activation parameters like mitochondrial potential, superoxide, ROS production using different mitochondrial dye. And we can see here that the blue one, when LSC supernatant was added, we found that the mitochondrial potential, superoxide, ROS production was inhibited. And here the blue one, the peak was shifted to the left. It means that these mitochondrial activation parameters were inhibited. It means that they are inhibiting mitochondria directly. And this is just within 2 hours. It means there is no transcription translation possibility. So they are acting on the mitochondria directly. So this was very novel finding in this work, where we have shown that the suppressive factor are acting directly on the mitochondria. Further, we extrapolated our finding from the mouse in vivo experiment and here to the human cancer patients. And for this, to test our hypothesis, we collected culture supernatant from different human cancer cell lines like A549, colon, and many, and we tested their effect on T cell proliferation in our established in vitro assay. And we found that some human cancer cell lines like colo, ES2, SCT15, PC3, SKBR3, they are releasing some suppressive factors that inhibit T cells proliferation. While on the other hand, some human cancer cells line like A549, DU, NCIH460, they are not releasing any kind of suppressive factor. So it means that our finding from the mouse are matching to the human population or human cancers as well. Now we will move to the next section, that is the strategy to rescue unresponsive tumors. Previously, we have reported that some uncouplers upregulate mitochondrial ROS, and that will actually this signal is very complex, but I have very simplified. So this ROS production will finally upregulate PGC1 alpha, which is transcoactivator, and that works with different transcription factor in this complex, like PGC1 alpha NRF complex or PGC1 alpha PPAR complex. And this complex will will upregulate many different factors and many different genes, and that will affect the mitochondrial biogenesis and fatty acid oxidation, oxidative phosphorylation, different pathways, and they will upregulate mitochondria. And we published this paper in PNAS with this title. In follow-up study, we targeted this complex, PGC1 alpha and PPAR complex, and we activated this complex with bijafabrate. Bijafabrate is a pan-PPAR agonist. So using this compound, we targeted this complex, and we found that the suppressive effect. The bijafabrate was with PD-1 blockade therapy. They have synergized, and the tumor was reduced very significantly, and the survival was increased. And we published this paper in Cancer Immunology Research in 2018. So based on our previous findings, these two reports, we tested the effect of bijafabrate, that whether they can rescue the suppression caused by LLC suppressive factor or not. So I added bijafabrate in the well where I added culture supernatant, and I measured the mitochondrial oxygen respiration, oxygen consumption rate, that whether they can rescue the mitochondrial respiration or not. And here the upper black triangle denotes LLC plus bijafabrate supernatant. And we can see here that compared to LLC only, the mitochondrial respiration was significantly enhanced. And here further we plotted some other parameters like basal respiration, maximal respiratory capacity, and ATP turnover. And we found that they were significantly a little improved. Means bijafabrate a little rescued the suppression from LLC supernatant. So encouraged with this our in vitro experiment results, I tested in the in vivo directly now in the mice. And first here we injected unresponsive tumors on day zero, the LLC, which is SIP positive, and B16, which is SIP negative as well as non-immunogenic. And we performed combination therapy and we measured the tumor size here. So the black circle denotes the combination therapy PD-L1 plus bijavuret, and we can see here that the tumor was a little reduced in of LLC, and even the survival was also enhanced. But the survival was not enhanced when B16 was injected, and this is expected because B16 is non-immunogenic, so they are not benefited by any kind of PD-1 blockade therapy or combination therapy because the immune system are not recognizing them. So this is the final conclusion. So SIP positive tumors release non-proteinaceous soluble small molecule less than 3 kDa size that inhibit mitochondrial respiration and proliferation of T cells, for example, LLC. And second, survival of host with SIP positive tumor can be improved by mitochondrial activation chemicals in combination with PD-1 blockade therapy. And this is a very simple cartoon that explains all the results of my project. Here, the cancer cells release some immunosuppressive small molecules, and that inhibit the mitochondria of CD T cells. And when we will perform this combination therapy, that is PD-1 blockade therapy plus mitochondrial activation chemicals like bijavuret, so the mitochondria will be enhanced, and the immune responses will be enhanced, and so we can rescue the less responsive cancer patients. And this work was published in eLife in March, and different science news portals were had covered this article, like this one, Eureka Alert, and many others. And finally, this is the acknowledgement slide. I would like to thank my supervisor, Professor Tasku Hunjo, and also different scholarships like JSPS, Takeda Science Foundation, just won us a scholarship. And these are some pictures that this is our PD-1 group. And we shared this moment when Professor Hunjo got Nobel Prize. That's all. Thank you for the listening.
Thanks a lot, Dr. Alok. That was a very insightful presentation.